Okay, good afternoon, everyone. Thank you for coming for the Science and Innovation session. My name is Kim Baldwin, and I'll be the moderator today. So we're going to start with Antonella Rivera, and she's going to be talking today about a fisheries monitoring dashboard for data-driven management. So very excited to be hosting the Science and Innovation section. Thanks for coming.
Gracias. Hi, good afternoon, everyone. Thank you for the ones who were able to come. Today, I want to present to you the work that my colleagues and I at the Coral Reef Alliance have been doing for a fisheries monitoring dashboard to promote data-driven management. So what if every co-manager and fisher could see how their fishery is doing live? This is Nelvin. He's a co-manager from the Tela Bay. He is the ED for ProLánzate, and they've been collecting data for the past 10 years on fisheries, fish landings mostly. And as he'll tell you right now in the video, you'll see that their main goal is to generate sustainable management policy. So it's not just data collection for data's sake.
Y hola amigos, mi nombre es Nelvin Bustamante, soy el director ejecutivo de la Fundación ProLánzate. Sí, como hablamos de pesquerías en la Bahía Estela, estamos hablando que han tenido mucha presión durante los últimos años en su parte marina como en la parte lagunar, ¿verdad? Debido a esta situación, ¿verdad? Los mismos pescadores que son los usuarios del recurso han tomado la iniciativa de acercarse a las autoridades, ¿verdad? Que son los que toman la decisión, que regulan el uso de este recurso pesquero para poder crear herramientas, ¿verdad? De manejo para poder hacer de que esta actividad productiva, ¿verdad? Se haga de una manera sostenible.
So you just heard Nelvin wants his fishery to develop these sustainable management policies based on that data collection. They have 10 years of data, and the Tela Bay is doing really well compared to other fisheries in the area. They get their results once or twice a year. But the problem is here, not that we don't have the data, is how to get them to the people who need them in an easy to understand way and fast. And this is a data action gap in small scale fisheries. We collect data, but sometimes it gets stored in a drawer somewhere or in a computer and it gets lost. Or the people who have the data don't know how to analyze it, or they don't know how to understand the results that are coming out, and everything gets lost in translation. And what this does, it leads to late decisions by the time we've already lost these species, or a lack of influence of the people who are really on the ground, like Nelvin or the fishers. And that is why we chose to build FishUp. With FishUp, we're trying to make data easily accessible, but what we're really doing is trying to co-produce understanding. And we're working as scientists with the fishers and the co-managers to design this information, make sure that we have very simple and guided indicators which are intuitive, that it's easy to access. You'll see now that you can see it on your phone or tablet, and that we build scientific capacity, but still maintain that scientific rigor. And because I want you to hear about the app, I don't want to go into much of the fishery science, but I do want to highlight that it does have a strong data backbone. We're using the standard land frequency data and analyses. We have data for multiple years and multiple sites, so over 25,000 data points for 6 communities in 10 years. We have standardized biological indicators for L infinity, length of maturity, etc. And we have these data sets that are compatible with existing data in the Mesoamerican Reef, which is where we work. So here's the dashboard for you guys to see it. It has multiple tabs. The first one is just a general description. Then you get a map of the areas where we're working and where the protected areas are. And you have this general summaries tab, which gives you all these small indicators and just information on the fishery, right? So what species are being caught, what gears are used, how much has been sampled, and you can select the time period that you want to see. And this general tab is basically what they were getting year after year or every 6 months. So we have both species-specific and multi-species indicators. We have these visuals, which are try to be easy to understand. We can still do better, and we know that. And it's based on the local stakeholder needs. So this was the poster that they would receive, and they would tell us, no, we need something different, or you need to move this around, or we need the dotted line to see when they're starting to reproduce, and that sort of information. So it's designed for fast, local decision-making. If we move to the next tab, which is the single species indicators, which is the ones that we're more commonly based with, what you can see is to the right, we have some very general, you can't see my thing, but to the right, we have some indicators, which are very easy to understand. But then we also have the graphs, which can be a little bit more complex for managers or scientists who want to go beyond. And all this information answers some easy or questions that they know that they want to get answers, such as, is the fishery being fished too young? Are there still breeders in the stock? When are they getting caught? And as I mentioned before, this is all based on standard length frequency data. So we want to make sure that the science is rigorous, but that the message is the one that's accessible to everyone. And once we've seen the single species indicators, we can zoom out to the multi-species indicators based on new studies that developed by Colding and Sheldon and Anderson on these types of size spectrum and life history trait models, where we want to integrate community level. So we're seeing how the whole community is doing, not just the top 20 species. And by the way, I forgot to mention, you can select what species you want to see in the single species indicators. We have the top 20. And here you can look at it at a community level and detect if there's an ecosystem change. So it includes aggregate information such as catch per unit effort, how much is getting caught with more or less effort. We have evenness and diversity indexes. Displaying. There we go. To see if it's balanced, are we catching just one species? Are we catching a little bit of everything? And then we also have a biomass spectrum slope, as I mentioned, with multi-species indicators. So what we want to do is, are we losing the fish or are we catching too many young in size? And like you just saw in this really short video that was recorded on a phone. So you can clearly see all this information on your phone as well. But what I don't want us to lose sight of is the human connection and that human component, which we're trying to highlight. So it was built by the people who use it and for the people who use it. The beta version that we're showing you now is from the Tela Bay in Honduras. It's a co-managed fishery, so the fishers are very active here, and that's why we're fortunate to work there. We tested this through an iterative process where they kept seeing it. We have descriptions, which you can click on and see the description in both English or Spanish. And we're all constantly formatting those descriptions so the fishers can also understand it. I don't know if we're quite there yet, but we've trained 10 NGOs in Honduras and 2 in Belize in data collection and in the data management and tidying process. That way we can have data for over 15 communities in the near future. And obviously, what we're already seeing is clear communication between scientists and co-managers. And hopefully, we can enhance ownership of these communities, and that way we can improve both compliance and power dynamics. So in the early wins or lessons that we're already seeing now is, as you can see in the map, it's been beta tested in these 6 communities, which are all located in an MPA. Yeah, and we're seeing that clear communication. So what do we want to do now? Where do we want to go beyond, and what are we looking at? So we want to go from this beta version that we have to a regional decision support tool that can help everybody who's here. We have socioeconomic indicators that we've also collected, so we want to include those in the app. We want to expand to other regions, so if everyone's interested. But we want to make sure that this is very simple. Like I said, this understanding is there. So we want to create a holistic fishery index, which can be some sort of stoplight that tells you how your fishery is doing, what range it is, and what management measures you can take. And in order to do this, we really need to work with a scientific network of specialists like the ones here to validate this information and develop that index. And finally, what we're seeing is ever since Monday in this conference is that there are a ton of fisheries dashboard and support tools. And we really believe that each has its own role. And we want to find a way to be able to integrate all these apps in a single way to get the information out there. I think we all have the same objective, right? We want to go from data to decisions, but we need to do it together. And we're looking to empower the communities to manage their data adaptively, obviously always based on science, and build this regional culture here of joint understanding. So if you want to join us, and this is something that you are interested in, here's my contact information if you scan the QR code. And I will be more than happy if you contact me to share with you that beta version that we're testing out. So you can give us your pointers, and hopefully at some point we can get your data out there as well. Thank you.
Great. That was an awesome presentation. Does anyone have any questions? All right, I do then.
Oh, sorry.
Thank you, Antonella, for that presentation. That was really amazing. It's really amazing to see how the organizations are connecting directly with the community. So I have a question regarding that. How do you get like the attention and the compromise from the community, from the fishers to be included in this process of co-managing, and how is the relationship with them in general?
Oh, yeah, thank you for that question. That's really interesting. And I guess it's not the answer that we want to hear, but it takes time. It's a long-term process. We've been working with them for a while. We have a 2021 paper on co-management as well. I think it's a marine policy. I can share that with you, where we looked at how the co-management process came to be. And it was a lot of years working directly with those communities. And as I said, it's very iterative, right? So you go in there, you show them the management measures, you show them the data, how their fishery is doing, and then we let them decide. So for better or for worse, they're the ones who are having the final decision. And we've seen improvements in a lot of fisheries, so lane snapper, and the blue runner, I think we're seeing how these species are going up because they're protecting them through co-management. But for instance, mackerel, which is like a bigger species, they chose not to establish minimum catch sizes. So we're seeing those populations go down. And what we're doing is part of this app is giving them that information so they know the results to their actions, right? That there are consequences to their actions. And that helps to get them more involved. So yes, I think it's working because it's a co-managed fishery, but what we want to do is work with all the local co-managers in the Caribbean who are already working with the communities so they can be that bridge between us and them to develop these types of tools.
I have a quick question.
Of course.
What about usability? Are you tracking who's using this app? Is it the managers? Is it the fishers? Are you looking into that as well? And which types of data sets they're using? Are they just using the broad scale, or are they actually digging down? That was my question.
That's a really good question as well. So we're in a beta version and not too many people are using it. Currently, who's using it, it's the co-manager. So we have Prolanza, Tenelvin, and Amatella in the Tela Bay, as well as the technical committee. We know that they're using it. And we are getting that feedback directly from them, what they're using. And we know that they're mostly using the indicators. Like that biomass spectrum can be a little bit too complicated. We're even considering taking it out. But what we want to do is at some point with the data and like the clicks, see what part of the app they're really using. So we're going to work on that. Thank
Thanks.
you.
Anyone else? Okay, so our next presentation is from Jennifer Loch. She's presenting virtually, so we'll be watching a short video about advancing fisheries science through accessible technology for community-driven reef fish monitoring.
In person, and I'm sorry that I couldn't be, but I'm happy to present about our SMILE project to advance fisheries science through accessible technology for community-driven reef fish monitoring. And first, I'd like to introduce our organization at Reef. We are a citizen science, nonprofit marine sightings database organization. And what that all means is that we have a worldwide membership of about 85,000 passionate citizen scientists that like to perform reef fish surveys. And so these divers are trained to do and participate in our hallmark project, which is the Volunteer Fish Survey Project. So on this infographic, it just highlights some of the successful numbers that we've had. But of our membership, 18,000 of them are active surveyors, which means that they are trained to collect relative abundance and diversity of fishes during their recreational dives. And they've submitted over 300,000 surveys in our 32-year history, which has contributed to over 134 scientific papers over that time. And so we do have a publicly accessible database, and with those information and with the metadata, it also reports if somebody is a novice or an expert surveyor. So we don't just throw them out into the water and take their word for it on what fish they see. These are experienced, trained divers and surveyors in fish diversity and providing those data. Traditionally, recreational divers are an underutilized source of data collection, but here with Reef, we can show how valuable they are to help fill data gaps in fisheries, especially in areas that may not be surveyed or with the frequency in which recreational divers are in the water. Of course, you all know, being in professional fisheries, that stock assessments incorporate size data for a variety of reasons. It can reflect reproductive potential, age, population change, things like that. These lengths are traditionally obtained through harvest or handling the fish itself, which of course can lead to a loss of that fish from the population through harvest, or it can induce stress if a fish is measured and re-released, like I'm doing in this picture with this ruler that's way too small. But this also can limit the amount of data that we have because of gear selectivity or fishing regulations. We often have a limited distribution of species as well as sizes that are sampled. There are existing in situ methods that can assist providing some of these data, but they are typically limited to scientists. They include things like the NOAA RVC method, which is a US federal agency that goes out and does visual surveys in its set area and time. And they use these T-bar sticks to assist them with visual estimation of lengths. These are highly trained divers, and this is, as you can probably imagine, a very resource-intensive source of data. There's also technology that we can use, such as the stereo video system shown here, which we use a calibrated dual GoPro system that uses event measure software on the back end to extract lengths from the imagery data. And then there's things like laser calipers that can work in a similar fashion. You can do this with the event measure software. You can do it in a more lower cost software like ImageJ. So the SMILE project was born out of the need to provide more data with our citizen scientists that are already collecting valuable diversity and relative abundance information by tasking them with now helping us get lengths in situ with a fun tool without ever having to handle the fish. So this is the SMILE, or Size Matters Innovative Lengths project, to use this handheld, easy to use camera to collect images of fish that we then use an AI workflow to extract those lengths. The goal, of course, is to complement the existing reef fish surveys, and with a tool that's easy to use and with a limited number of species, so we don't have people that are intimidated by learning all the species in a region. Ultimately, these data will be publicly available for our end users especially, so hopefully that can improve future stock assessments, especially for data limited species, and ultimately inform management and conservation efforts of those species. So with that being said, this is our curated list of target species. So again, we don't want to task overload our citizen scientists, but we also want to get critical data. So we ask them to work with this list of fish that we selected based on their ecological and economic significance. So we ask them about any large grouper basically that they encounter, but in our region, the 4 common ones are black, Nassau, red, and Goliath groupers, as well as hogfish, 3 species of snapper, mutton, yellowtail, and gray or mangrove snapper, and then 4 species of parrotfish. And these parrotfish we use more as an ecological indicator since they are herbivores or corallivores, and they give us a depiction maybe of what's going on in the reef from that trophic level. So we have midnight parrotfish, rainbow parrotfish, blue parrotfish, and the ever so common stoplight parrotfish that is available in 3 different phases. And so we ask our citizen scientists to take pictures of any size that they encounter so we can improve our size distribution of these species. The tool that we use is the Fish Sense Light Camera, or FSL for short. This involves using an Olympus TG6 or TG7 camera that is contained in its typical underwater housing. And we have a detachable wide-angle lens or dome port, which is used to reduce the distortion on the images used in post-processing. And then we have a cold shoe mount that holds up a single laser. The way that we have our data collected is we've been focusing in the Florida Keys, where the Fish Sense Light camera is used by citizen science divers to collect images of these 12 species. First, these divers are given a brief online training even before they arrive at the dock, and then some dockside instruction on how to use the camera itself. The camera does have to have certain settings, such as having the raw file associated with each image, so that setting is on. They can't use the flash. And they have basically like a list of typical settings for the camera itself. And then we shoot in burst mode so that we can capture lots of images of a particular fish. So we have some choices to work with when we're extracting our lengths. Then we ask the divers to perform their dive. So first they have to take some images of a dive calibration slate rather. And they do that at the end of their dive as well. Then we ask them to take images of fish with the laser on the center lateral side of it, as you can see in this image of an intermediate stoplight parrotfish. And then they just go about their dive as they interact and encounter any of these target species, they'll take pictures of it. And then we will extract those links through a post-processing AI workflow. These have been compared to stereo video cameras used by staff, so we can help validate these methods against an existing technology. For post-processing, for the sake of time, I'm just going to kind of touch on this, but the laser gives us an indication of how far away the subject is from the camera itself, so that we know based on depth of field that things that are farther away seem smaller even though that they're not, or things closer up seem larger even though they may not be. So the laser gives us a distance between the camera and the fish, and that gets incorporated into the calculation of where the head and the tail are in that image of the fish. And this is why the raw file is important. So each of those positions has an XY coordinate associated with it. We know where the laser is expected to be. So at different distances, that the laser dot, if that fish was further away, the dot on the laser would be in a different position and thus be, influence the calculation in that way. I'm just going to touch on our citizen science participation, because we can probably do a whole other talk on this, but we've had 50 different volunteers that have come out to work on the project. A lot of volunteers actually repeat. So I've had, more volunteers come out, but unique volunteers. We've had 50 of them on 295 total dives in the Keys, along with 9 reef survey trips. So these are trips that are taken in other areas of the tropical Western Atlantic, such as Cuba, Bonaire, Belize, Curacao, the Caymans. These cameras are way more well-traveled than I am. This is not me taking them, it's other people taking them out on these trips. And then we're actually scaling up this project to have local diver operators be the ones to kind of get their clientele interested in this conservation initiative and getting them used to kind of using the camera, having a shortened webinar that they can use in the dive shop or as a refresher for people that have been engaged via email before the dives, that kind of thing. So in this next phase of the grant, we're working out how to best the best methodology for involving dive shops to do that. And then we've developed a formal survey that could also be the subject of its own talk, looking at motivators and barriers to participation, as well as the project itself. So getting back to the length question, looking at some preliminary results for length between the 2 camera systems. So on these figures, it's a panel that's all laid out the same, where on the x-axis we have fork length, y-axis you have frequency, so how many individuals at that length. These are length frequency distributions, nice little curves. And then the stereo video camera, that professional calibrated dual GoPro system, is in blue, and the Fish Sense Lite, this novel camera system for the Smile project, is in tan. The vertical dotted lines are the means for each of those camera systems. So in the upper left figure are length frequency distributions for all fish collected across 4 dives. And what we see is that there is a slight difference in the means, that the stereo videos are slightly larger, but there's no statistical difference. And this trend holds up as you go across the panel, but the results might be a little bit more dramatic because of lower sample size, and that's not surprising. So as we look at midnight parrotfish and gray snapper on panel B and C, fewer individuals were measured for these dives, but we see that there's still no statistical difference between their lengths between the 2 camera systems, although stereo video does still measure larger. We shrink the differences in those means as we improve our sample size. So we have hogfish in panel D. Stoplight parrotfish are our most common species, so those 2 camera systems get even closer with them. Same trend, no statistical difference. Stereo video is slightly larger. And lastly, leaving off with blue parrotfish in panel F has a similar trend as well. Looking at the regressions between these 2 camera systems, we have a Fish Sense Lite on the x-axis and stereo video on the y. These are their max lengths, and there's a lot of agreement between these 2 camera systems as well by looking at it from this perspective. And you may be wondering which camera system is right. Well, the way that we get at this is by looking at known objects. So here we take a fish model, a fake fish underwater, which may be a little confusing to the live fish on the reef. And we look at it by camera type. So we have 2 stereo video units here in blue, 1 Fish Sense Lite unit. We actually have 11 of these cameras. So we'll be processing not 11 cameras per se for this, but other camera units to build out these estimates. But we have mean length on the y-axis, and the dotted horizontal line is the size of that particular object. You could see how close these camera systems are to reality for the known size of its object. It's within the same range that anybody would have measuring fish on a fish board. Stereo video is still slightly larger here and still no statistical difference between the two systems. Just kind of highlighting some of the things that we can do with these data. These are some length frequency distributions across species. The left-hand side has the more common species for this project, the mangrove and gray snapper, and these stoplight parrotfish. On the right-hand side is a glimpse at some of the more rare or less common species that we encounter, basically the grouper and snapper. Hogfish are fairly common. They can be spoken of in their own right. But just kind of looking at some generalized linear models for now with our fixed effects of site status, so being a protected area or outside of that, depth, date, and species. The leading model for predicting length is actually the site status, so in a protected area or not, as well as max depth. So a lot of the non-protected areas we looked at were also in the deeper area that they are looking to expand the protected area. And so just to highlight some of this, not only do we not see some of these species in our surveys on the deeper non-protected areas, but a lot of the species, most of them are larger within the protected area. And so that gives us some support that protected areas are working. And it would be great if we can expand that no fishing area to these deeper contours. I just want to mention too that part of this project is providing publicly available data. And so we have developed this data display with our partners at Socorro and Axelle. And we have 2 layers on this data visualization. It goes across the Caribbean, but our SMILE data has really only been focused in the Florida Keys for now. The SMILE data layer looks at the length data that we have and number of surveys in the past 2 years of the project, and it will be continuously updated. And similarly for Reef's Volunteer Fish Survey Project layer, we have sighting frequency and the number of surveys, and that goes from 1993 until present. And so some of the things that when you're looking at this data display is you can even turn off one or the other of these layers. You can toggle all the species on. You could select one species, a few species if you wanted to look at all the grouper. Down on the left-hand side of our panel, it gives you a actual like data visualization of the quantitative data. And you can do that within a broad area. So you can zoom in on each of these hexes and look at data just in that particular area. And you can look at some other metrics like median length and minimum and sighting frequency. So it'll also have the reef fish survey information within that hexagon as well. These data will also be publicly, well, I should say they'll be accessible for the raw data upon request. So if there's a region that you're interested in looking at, or you want to look across the broader region for where we've sampled for SMILE data and the Volunteer Fish Survey Project as well, you can make a request to us and we'll give you the data as well as the metadata associated with that. So we look forward to sharing this and seeing these data put to use. So in summary, our camera systems are comparable to each other to generate lengths. We've had a positive citizen science response thus far, and we will have these data publicly available similar to Reef's Volunteer Fish Survey Project. And with that, I want to thank our project partners that we collaborate very closely with at the South Atlantic Fishery Management Council, Scripps Institute of Oceanography, and the Engineers for Exploration at UC San Diego, Socorro, Tetra Tech, and Axiom Data Science, as well as our funders at NORA Coral Reef Conservation Program. And with that,
Okay, you can reach out to her if you have any questions. We're going to keep moving along here. So the next presentation is by Sander VanLopik. Hopefully I said that right, Sander, where innovation meets the reef, tech tools for conservation.
Yes. Hello, my name is Sander VanLopik. I'm the founder of Rougher Reefs. I'm a field technician for the program, and I'm based in the Netherlands. And it may sound weird, but I work in a zoo, but I'm full-time conservationist, and I will explain more about that during the presentation. And what is Rougher Reefs? Rougher Reefs is a program that looks on coral reef fish, and it's to understand the reproduction and their early life stages. And literally in the Caribbean, there's not much known. Before we started 3 years ago, we could only find 12 colorized pictures of pelagic fish eggs. And for Bonaire, we should get up to 300 because there's more than 350 fish species on Bonaire. And I'm really happy to tell you that we're now already at 30. So basically what I always explain if I'm telling about my job is, first of all, I'm not a researcher, I'm an inventor. So we have researchers doing the work. And I really try to be the same kid that looked at frog spawn when I was little. So I always say, what we have is Kinder Buenos for adults. Why focus on the reef fish? Well, we actually know that they're really important, but we can't do too much about it. So managing these stocks, except of not getting them out, is basically the only thing that we do. But I think we can do that differently. And we have some steps in there to actually get there. Because if we have no reproduction of fish, then we also wouldn't have coral reefs. And it's called coral reefs. So it's not only about corals, it's about the residents of the reef as well. And we work with a 3-pillar approach. So basically, we have a breeding system. That's our nature-based solution, where we try to see if we can also reproduce fish in open waters. So basically, it's an UFO, an unidentified floating object, where we try to see if we can hatch these eggs in the water. We also have our science-based pillar, because of course you want to prove that what you want to achieve is actually working. And we have the local-based pillar, where we actively engage with fishermen. And I always say we do this together. It's maybe my idea, but I can't do that stuff on my own. So we do this with, for example, the fishermen of Piscabon on Bonaire. So we are always on an expedition. We do this literally every day. And what we always do is we place nets in the water in the afternoon. We hang them overnight, and in the morning we collect them. And we have these bottles. We take them to the lab, try to do some photo ID, then figure out it won't happen that well because we only are at 10% of the 100. So we also need to do DNA samples. And in order to do something with the data, it's really hard because if you are only at 10%, we need to make sure that we can get it faster, of course. So we made something that is very cool, I think. This is our artificial intelligence tool to see if we can make a DNA sample or not. So basically, all the dots that you see over there are pictures that we ever took from a fish egg. This is a little bit older, but we have way more. So we're almost up to 10,000 pictures of different fish eggs now. And when we have a DNA hit, we give it a color. So basically what we do is in the file drop, we put the picture in. We can move around and, how do you say? Get up close to all these dots and then see if we already have references via the AI. So we know that we should not take a DNA sample. Otherwise, we would sample the same eggs again and again and again. And it works quite well. So, for example, you see on the left side, you see a small patch that is all of the parrotfish. So we can actually see that if it recognizes a species, then it's getting better and better at it. And yeah, it's really cool to see that something that started 3 years ago as an idea, where I was literally with my mountain bike and 2 buckets on Bonaire to show that we were able to catch fish eggs, is actually getting as big as we would hope. But of course, this is not the end goal, because what we want to see is if we can also put them back into the water, because we have this hatchery system. But there's one other cool thing that we did, because what actually happens if we don't catch fish jizz? Sometimes you don't know. Here you can see one of the spawning grounds that we found. So every single day, the yellowtail parrotfish are spawning. And what we did is try to see if with a particle tracing model, we could see where it ends up. And the fun part is within 24 hours, it is still in open waters. Well, we know that the yellowtail parrotfish if they are hatched from the egg, should end up on the reefs, which means that basically what we want to do is see if we can make an application. Surface currents are wind driven. So if you know that the wind is in the right direction and nature can help itself, why would you do something for conservation? You can also switch that around, of course. And the cool thing about this is that with parrotfish, because we know they spawn every day, we can literally do this every day, these testing. And if we work on the breeding system, it's actually only for the first life stages for now, because we don't want to make the same mistakes as in aquaria, for example, because everything we do there is try to see if we can close the full life cycle of the fish. And close a full life cycle is very hard. Of all the fish that were ever kept in captivity, only 15% has ever been reproduced of the species, which means that if you walk through an aquarium, you most of the time see only fish that we can reproduce. But for example, for these two species, we don't have any references because there's never been bred in captivity before. And we can see that with certain fish, we're already getting there quite well. The survival rate is getting higher and higher, but with parrotfish, also a species that's not been kept in captivity that much, we can see that it's already still a bit harder to breed them out. But these numbers are actually quite high. Does anybody know how big the survival rate within 24 hours is of most pelagic fish? Pelagic spawners? For example, an Atlantic herring, only 2% survives within 24 hours. Sometimes within, because these are the results after 3 days, sometimes in 24 hours with the parrotfish, we still get a hatching and survival rate of 50%, which is actually quite nice. Because then in conservation numbers, you can do something that's never been done before. And of course, we need to do a lot of more testing and see if we can get further with it, because there's a lot of things uncovered here. Everything that we know from land-based facilities needs to be adapted to the sea. And it's really a hell of a job, but it's very cool to do. So one of the things that we are currently looking into is how can we make sure that there's less energy spending on swimming, because we can actually see if there's too much movement in the water, then there's less, there's a lower survival rate. But we are getting there, and literally nobody's ever been there. So even the mistakes are still good, because actually we need to learn something of these first life stages of fish. So we're still further optimizing the survival rates. And the next step will be to see if we can release larvae in large amounts and then trace back what is actually happening if we do this. As I say, a lot of stuff here is uncovered, so really curious where this will end up. For the next phase, we also thought, how can we improve the breeding system a little bit? So we were upscaling, we were thinking about upscaling and also a more ecosystem approach. Because of course, it's also not only about fish. It's about fish, corals, sea urchins, and maybe even more. And the cool thing that we have now is we have a light diffuser within the breeding system. So underneath the larvae trials, we can also do coral growing trials. These trials will start within Burgershoe, the biggest coral reef aquarium that is not connected to the sea, next year. And of course, we should also not forget the local pillar. Last year we presented it. Fish are not only friends, they're also food. We should really make sure that we get in more depth together and see if we can actually also reduce what we take out of the water. And one of the coolest ideas that I think we had in the last year is see if we can maybe do something with the fish waste of the fishermen. For example, in the Netherlands, in terms of fish meal, there's already a lot of fish fed to dogs only. I made a calculation for the United States. And in the United States, there are 75.8 million dogs, which means that in wet weight, to dogs only in the United States, we feed 200 million, 2,000 million kilograms of fish yearly. So I always say, should we not also think about our pets, about the carbon paw print? Because the love for, for animals doesn't stop at the animals at home, of course. So one of the things that we're looking into right now is see if we can use the fish waste of the fishermen to see if we can make pet food with it. And we did the first test last month on Bonaire, and it actually worked quite well. As you can see, the dog is really begging for it. I think if we have burning hoops, he would also jump through it. So that was really already a nice first start. And it's very cool because literally, in terms of conservation, most of the time it's about what we're putting back, but it could also be about reducing what we get out, because there's really a lot of wind to there. If you have any questions, feel free to ask them. In the meantime, you can enjoy the very cool video of a parrotfish getting out of the egg. If you look this up on YouTube, you still can't find it. If I have a shitty day, I always watch this because this is super cool. The hatching of the Kinder Bueno for adults.
Great presentation. Do we have any questions? All right. Okay. I give the 3 minutes. We have 3 minutes, yes or no? Do we have 3 minutes, yes or no?
I was in time.
Yeah, that's what I
Now
thought.
everybody's
We didn't
wishing the
count
parrotfish
three.
hatching.
Okay, we do have time for a quick
Yes.
question.
Thank you, Sander, for the presentation. It was very interesting. There was so much information there, but you made it very visual, so I really liked it. I want to go back to the beginning where you were talking about AI to identify fish eggs. I feel like that is very interesting and that can be used in a lot of different areas as well. So I was wondering, how are you training this AI to know what species it is, and how many samples do you need to get that training up to par where you need it to be?
So here it's getting funny because we figured out for the Caribbean it's really hard because there's not much known of Caribbean reef fish. So I figured out, let's go back to the place where I started. So now we're starting a program in the zoos. It's actually a breeding a program before breeding programs, like we have for pandas and stuff. So the idea is to see if we can actually empty out the database for every multi-species tank. And then, for example, in reef tanks, we already know which species are there and then fill it up again. So we have a better learning curve with yes and no questions. Because AI, of course, needs to be trained. And we can see that if we empty it out and we already know which species correspond to which egg, it's getting faster. So we want to do that first within zoos. And then that's also a very cool way for the zoos to contribute to conservation and ocean knowledge, because literally sometimes we forgot to measure new life and thus hope for the coral reefs.
Okay, we have time for one more question, quick one? All right, thank you, Xander. If anyone has questions, see you back there. Our next presentation is from Hadrien Bidenbach. Hi. So One Shark Integrated Approach to Tiger Shark Conservation, Risk Management in the Caribbean.
This one. Yeah. All good? Yeah. Okay, so hello everyone. My name is Hadrien Bidenbach. I'm working for the One Shark company. I am the director and also the PhD student. And today I will present you a possible integrated approach to tiger shark conservation and risk management to the Caribbean. So just to add a little bit of context, the first question that the government asks after a bite event is what will be the consequences. The first consequences will be a big fear with the people nearby the sea. And the second one will be economical consequences, mainly about the tourism at sea, for obvious reasons. And so that's what happened in Sint Maarten on the French side of the island. In 2020, we had a fatal bite, and then a few weeks later, in St. Kitts, the island in front of Sint Maarten, we had another bite. And that's what created the initiative to develop the One Shark company. And our main topic are the scientific research, the awareness to people and institutions, and to develop a sustainable shark risk management. Because there is two opposing views on risk management between the terrestrial environments and the marine one. In the terrestrial environment, it's common to do selective management to remove the problem individual that kill people. But on the marine ecosystems, mainly on French island and other ocean, it's common to do non-selective management with shark, for example, and to do indiscriminate removal of the entire population, even though these predators share the same similar concern. That's why we try to develop an innovative approach with the shark risk management, assuming that it could work like on the terrestrial environment with a problematic individual. And that allows us to question the regulatory fishing efficiencies. Because on some place we saw that the regulatory fishing were extremely efficacious, but not on other. And on the picture on the right side of this, you can see that if we fish randomly the yellow shark, non-dangerous shark, nothing will happen. But if you catch the red one, the bad one that bites, for example, we will have a straight improvement on the risk management with a less number of victims annually. And we show that it could work by swabbing the wounds of the people that were bites in 2020 in Sint Maarten and in St. Kitts. And with genetical analysis on the microsatellites, we were able to attribute these two bites to the same tiger shark. So now, with my PhD, the main question is how to optimize the chances of detecting a biting animal. First of all, to know, we need to know where are they, how many are there, and how do they use their environment. So for that, we do non-lethal fishing data collection. It's just using floats with an anchor and a barbless hook with a rotten meat to select only the tiger sharks. And with that, we create fishing-related data with the GPS position of the line, the type of bait, etc. Biological-related data with the DNA sampling, the tagging parts of the protocol, and also we do an additional data with, for example, the regional bathymetry to add covariables. So on the tagging parts, first of all, we do the DNA sample by doing some notches on the dorsal fin. It's helped to shape the dorsal fin and to facilitate the photo identification, and also to take a genetic sample. Then we put permanent plastic tag on the back of the dorsal fin. They are all unique with a unique color barcode to facilitate the participatory observation, because with that, we are able to identify a shark from a distance with only a GoPro. And then if the sharks are big enough, 3 meters plus, we deploy a GPS tag to track them on the regions. And with all of that, we develop a network of collaboration involving institutional scientists and marine professionals spread across 9 different islands in 5 Caribbean countries, and we continuing to develop this regional cooperation. So the first preliminary results on the chapter 1 were there. That's the CPUE map of the fishing area in Sint Maarten, Anguilla, and Saint Barts. And in 3 years nearly, we dropped 219 line and catch only tiger shark on the hook because of the specific bait we use. And we were able to see that the closer we are to the drop-off, in average, the shark are bigger. And on this map, all the data are from the same satellite tag, so the same individual animal. And we saw that the shark was crossing all around the region of the Lesser Antilla and going on nearly all the surrounding islands of Saint Martin. So that's also explain why we need to cooperate between each other. And on the right side, you can see the depth history of this shark at the top. And we saw that he spent lots of time at the surface between 0 to 40 meters. And he also do lots of traveling straight to the open sea and then come back to the shore. We can see that on the graph at the bottom right. How do they use their environment still on the same data sets? We were able also to see that the warmer the sea is at left, the deeper the shark goes to cool down a little bit. And on the right side, you can see in this graph the proportion of time between depth according to the distance to the coast to highlight where the shark spent the most of his time during 6 months. And he spent all this time between 0 to 20 kilometers of the coast and at around 20-meter depth. And these data are really useful because we could implement them in conservation or risk management issue. First of all, conservation to, for example, to give some advice on how to develop marine protected areas specifically for shark, for tiger shark, or to help government that have a shark issue to advise them how to recatch a specific individual. And then the last chapter of my PhD is how many are they? So in 3 years, we did around 100 samples with no recatch on tiger shark around Saint Martin. And so we will be obliged to do a genetical analysis to determine the shark population size. So for that, I did the extraction and amplification of all my samples, then the bioinformatic, and now I'm doing the closed skin mark recapture analysis to develop an accurate number of this shark population of the Lesser Antilla. And by shark population, I'm working only on tiger shark. And so finally, how we would like to cooperate regionally. We would like to develop large-scale data sharing to optimize the resource of everyone that drop at sea, and also to increase the coverage and the possible scope of the scientific publication related. Then we will need to have a regional cooperation to centralize all the DNA samples that are collected in the area and to help us on the recovery of telemetric tool at sea. And also we could help in an event of a bite somewhere to share our swabbing protocol, emergency protocol, and assist with the One Shark emergency network that you could reach if you send me an email, and I will add you straight on it. Thank you for your attention.
We've got time for a question. All right, it looks like we don't have any
Okay,
questions.
great.
Fine, we'll keep moving.
Yeah,
along.
thank you very much, and don't hesitate to contact me at the lunch break.
All right, so next up is Tadzio Bervoets. Sorry. Advancing a Caribbean sustainable blue economy, development of a regional blue finance hub and implementation of sustainable finance mechanisms.
Thank you so much. And you got my name about 85% correct. I appreciate it. Good afternoon, everyone. My name is Tadzio. It's a pleasure to be here. I'm not so sure if GCFI has realized, but they put me right behind two close collaborators of mine, which is quite interesting. But I'm going to shift gears a little bit. I am going to talk about advancing the Caribbean sustainable blue economy through the Carib Bluefin project. And I am Tadzio, as I mentioned, and I am the project lead on that specific project. So I'm going to talk very briefly about the Caribbean Biodiversity Fund, CBF. So the Caribbean Biodiversity Fund provides sustainable financing for projects within the Caribbean region to protect the lands and waters on which all life depends. The way we do this is through ensuring continuous funding for, this is small, for conservation and sustainable development in our region. Our vision is a Caribbean region where both the natural environment and communities are conserved. You can see the beneficiary countries that CBF works with here. And I must mention that I failed to remove one of our donors that is no longer a donor, USAID, for obvious reasons. A little bit more about the CBF program. There are three programs of the Caribbean Biodiversity Fund. The first is the Conservation Finance Program. The Conservation Finance Program specifically provides financial resources through partner national conservation trust funds to manage and protect biodiversity in 11 regional countries. And for the context of this presentation, it's important to note that the revenues generated by the financial mechanisms, which I will mention subsequently in some of my next slides, will be channeled through the National Conservation Trust Funds. We also have a climate change program, which works through the Ecosystem-Based Adaptation Facility. And then we also have a Nature-Based Economies program, which works through the Advancing Circular Economies Facility. In terms of the Carib Bluefin project, it's a 4-year project. It started in April 2023. And the specific objective of the project is to create and strengthen nature-based blue economy opportunities and approaches in the Caribbean through innovative financial mechanisms and the development of a blue economy hub. The project will also engage stakeholders and raise awareness and sustainable blue economy within our region. It is funded by the Global Environment Facility, implemented by the United Nations Environment Program, and executed by us at the Caribbean Biodiversity Fund. How will we implement public-private partnerships? Although one of the most complex components of the project is also one of the most critical in ensuring that sustainable investments and integrating the private sector into the development of financial mechanisms is done efficiently. And it is also aligned with the broader environmental and economic policies within the region. This project specifically positions the blue economy as a catalyst for sustainable growth, promising a future for prosperity and environmental stewardship in our region. In terms of project scope and components, component 1 are the financial instruments. So the objective of developing these financial implements is to implement financial products and services to fund sustainable marine and coastal projects. I will talk a little bit more about the specific financial instruments during some of my next slides. And the outcome is to increase accessibility and mobilization of financial resources for sustainable blue economic initiatives. In terms of the financial instruments that we have been developing, or the financial mechanisms, if you will, we've been working on developing a digital payment app, so a mechanism that enables digital fee collection for access to marine protected areas with revenues allocated to conservation and sustainable management projects. And again, it's important to realize that the channeling of this funding will go through the National Conservation Trust Funds in the project countries. Then another very complex component of the project is developing a blue carbon facility, a facility that provides technical assistance and financial support to blue carbon projects within scalability potential and the ability to generate returns through blue carbon credits. Then we're also working on Blue Card, whether it's a credit card or a debit card. But the idea is that we're working on a facility whereby the purchase is made with a credit card, the percentage of which will go towards conservation and sustainable development activities. And I wanted to pay specific attention for obvious reasons, considering what we've been going through in the region on the Reef Risk Insurance Facility that we've recently launched at CBF, which is basically a financial mechanism that protects coral reefs from damage caused by hurricanes or other extreme events through a parametric insurance policy that is triggered by the severity of the event and releases resources to implement rapid reef restoration and conservation actions. So this specific mechanism is called the Reef Resilience and Risk Financing Mechanism, R3FM in the Caribbean. The project goal of the facility is to develop and implement an effective, efficient, and sustainable regional reef insurance program to reliably finance post-hurricane reef response in the Caribbean, thereby improving the resiliency of coral reefs and the vulnerable communities who rely on healthy reefs for livelihood, food security, and coastal protection. This is a meso-level regional reef insurance, parametric reef insurance program that focuses on planned reef response, starting in initially 4 pilot countries, so Jamaica, the DR, St. Lucia, and SVG. And we're really focusing on building local, national, and regional capacity and preparedness, which includes developing operational procedures and financial processes to implement the parametric insurance scheme. Also, the designing and structuring, pricing, and placing the reef insurance program, and then most definitely creating the conditions for long-term continuation of the reef insurance facility. So in terms of output, building local, national, and regional capacity and preparedness, we're really building up on local demand. We're also working on a very efficient system for rapid payout and distribution. We're also working on developing reef response governance and plans, reef response budgeting. And one of the interesting parts of this project is that we're also working on developing reef response brigades that we can mobilize to go into the communities right after a major weather event to ensure that restoration work is done efficiently. And then, of course, we have significant focus and emphasis on inclusion and societal protection. In terms of designing, structuring, pricing, and placing the reef insurance program, we've been working very closely with the Mesoamerican Reef, with the MAR Fund, who also has a similar parametric insurance scheme, in developing a trigger mechanism. This is focused on a bespoke payout structure based on reef response plan at each covered site and funding needs at different event intensities. So category 1 triggers a specific payout all the way up to category 5, which also triggers a specific payout. And in terms of output 3, creating the conditions for long-term continuation of the reef insurance program, we're working on awareness raising, network of allies and champions, development and rollout of Caribbean Ocean Finance Mechanism, a sustainable finance roadmap, and active fundraising, sustainable premium financing through the endowment. Of course, we're not operating in silos as part of the program. We're working also with other projects within the CBF. So we're working together with the Crab Project, has nothing to do with crustaceans, but is focused on the Caribbean Regional Architecture for Biodiversity. And they will specifically focus on training of the reef response brigades. My project will mostly be focused on ensuring the sustainability of the program, especially through premium financing from the revenue generated by the financial mechanisms. And the core project, the Caribbean Organizations for a Resilient Environment, will also focus on sustainability and fundraising through their project. And then just to move quickly to the Sustainable Blue Finance Hub, which is also a component of the Bluefin project, we're establishing a centralized platform to support the growth of sustainable marine-related businesses and startups. We are also creating a digital hub that provides information, resources, and network opportunities for entrepreneurs, highlighting investment opportunities and success stories. And hopefully the outcome of that will be the promotion of innovation and entrepreneurship in the blue economy, facilitating knowledge exchange and business development. Of course, knowledge sharing and replication is critically important because we want to spread this throughout the region. So we're working hard on disseminating knowledge and best practices, developing case studies, conducting workshops, et cetera, and then through that, strengthening capacity and increased awareness among stakeholders, leading to improved practices and policies for marine conservation. Just to get a little bit more granular in the Caribbean Blue Economy Hub, some of the function of the hub are to facilitate connections between investors and blue economy projects and businesses through a clearinghouse mechanism, facilitating networking through a regional blue economy working group, which was established about 3 weeks ago. We have some members of the working group in the room here with us. offering training and capacity building through webinars, courses, key resources that are available regionally, and then, of course, providing educational resources on entrepreneurial training and blue economy opportunities. Some more features of the hub, as I mentioned, the clearinghouse mechanism, the development of the technical resource library, providing opportunities for training and education modules, networking opportunities, which is also critically important, connecting with experts, and for people who are somewhat technologically challenged myself, we're really putting strong emphasis on ensuring that the hub has a very user-friendly interface. And that is my talk. Thank you. Happy to use 1 minute and 15 seconds for questions.
We definitely have time for a couple questions. I thought I saw a hand. No. I have a question.
Yeah, I knew you would.
I just had a question about the hub, and I think that that's great. I think one of these solutions, and I think we talked about it in our session, is highlighting innovative solutions that are working. My question, and this is my personal thing, the blue economy word is thrown around left, right, and center. Have you put in or built in mechanisms to check or monitor the evaluation of these organizations that say and monitor, the management effectiveness and accountability that they're actually doing? Because if you're getting investment in.
Sure. So we've built in a very robust MENL framework, so management and learning frameworks. But especially through the working group that we established, they are sort of the vetting mechanism for projects, organizations, initiatives that will be hosted on the hub. So we do have a process in place to ensure that the projects that the hub supports, that they are properly vetted. And in terms of terminology, because we're a GEF-funded program, we usually, or we do use the World Bank definition of the blue economy, which we use as sort of the basis for the premise for departuring and how we frame the discussions around blue economy and how we support those initiatives in the region.
Great. Any other questions?
That's it. Thank you very much.
Okay. All right, so next is Shelly-Ann Cox from my home country of Barbados, transforming fisheries through data innovation, building the future of sustainable resource management. And so this is a virtual presentation, but I talked to Shelly-Ann. She is online with us, so she will be providing time to host questions. I believe her presentation is just under 10 minutes, so great.
My name is Christian Payne. I am the CEO and co-founder of Intelligent Technology, Inc. You seem to have gotten my clicker. All right. So about a year ago, we started collaborating with the Barbados Fisheries Division. Today, I will tell you about some of the things that we would have learned, some of the challenges that we would face, and show you some of the things that we've built. But before I do that, let's talk about some of the challenges with the legacy system. As Jalissa just talked about, when we started collaborating a year ago, the Fisheries Division was using a lot of disparate tools. What this meant is that while they were using all these different softwares like Pogo Toolbox, Excel sheets, and different sets of software to actually do this analysis and provide these services to the Barbados Fisher folks, as well as providing their analytics to their different stakeholders like the Barbados Central Bank and the Barbados Coast Guard and whatnot. This obviously created 4 core problems. The first thing is that the technology itself, while it was fantastic to run the Fisheries Division, going into our new age, which is focused on AI agents and all these types of technologies, it wouldn't have scale to meet the needs. The second thing is, as I mentioned before, and as Jalissa mentioned, this data was fragmented. It was stored in so many different places that if you wanted to do analysis, most of your time was spent in what we call data engineering, i.e. sitting down, figuring out how you're going to connect these different data sets to be able to clean it and then do your analysis. And the other thing is that that's just completely inefficient. And then finally, most importantly, this data was still only had limited access, i.e. it was only available to certain stakeholders. So for instance, Jalissa obviously would have access to it and maybe the Fisher folks, but if other people wanted access to this data, like researchers, it would become a more cumbersome process to actually get access to this data. So what do? Well, us and the team at the Barbados Fisheries Division sat down and thought about FAS. What is FAS? Well, firstly, it's going to be a centralized cloud platform, right? It's API first architecture, meaning that it's able to communicate with all software platforms across the internet, i.e. if someone wanting to offer services to a fisher folk, they will be able to integrate with the Barbados Fisheries API to be able to offer those services to the fisher folks. If other technical authorities within the Barbados government wanted to be able to pull data directly, like the Coastal Management Zone or the Barbados Coast Guard, they'll be able to pull this data directly from the Barbados Fisheries Division with no need for being able to, no need to always have to contact them directly. The second thing is that we started pulling all the different data from the different components. So VMS data will be in the platform, data related to fish landings, as we'll show you later, data related to the inspections. All these different data sources that which were previously fragmented are now being centralized and combined into one place, as well as markets analytics. So let's talk about a bit what's under the hood. So firstly, everything is built on open standards. Most of it is open source. So even down to how we store the geographical data, everything is built on open API standards. Essentially, these APIs are well documented and easily able and accessible for anyone to be able to use. The second thing is that our infrastructure is serverless. So while we do have servers that will serve you web technology, we don't actually maintain a physical server, even though we will have one for backup purposes and data backup purposes. The second thing is, as I mentioned before, everything is API driven. This may not mean anything to somebody to non-technical folks, but to give a better example of this, it will allow us to ensure that we can control who sees what data when they're pulling the request. It gives you full control of your data governance and having this direct ability to actually be able to scope permissions. And the final thing is our top layer, which is our analytical and AI side. So the reason and the rationale behind doing everything API first is because in this coming time, we're going to be building more agentic technology. We're going to be using AI heavily within the actual fishery sector. As Dr. Cox said, we're going to want to do image processing. We're going to want to do more and more interesting things. And to do that, you have to have a very, very solid foundation to build on top of. So I'm going to show you, is this going to play? This is supposed to, oh, okay. All right, so this would be the new data collections form outside of Cobalt Toolbox. It works fully offline. I'm going through it really quickly, and it will collect all the data. It's mobile friendly. And after you put in your observations, you go straight to your fish landings. You get a nice platform that shows you all the data. And this is the fish landings dashboard that Dr. Cox was talking about earlier. This is the first version of it that we'll be excited to start debuting. So this is our analytics homepage. Here, you're going to see the different dashboards that you have. We're going to start with seafood inspection. You're able to see your total inspection, your pass rate, and your pending results, as well as the weekly inspection results. You can see your quantum metrics, your species inspected. You can also see your recent inspection results, as well as any the different facilities at which the processing is happening. And then we're also trying to bring in the cold chain monitoring. Right. So going back up, right, we're also allowing to see the repair and maintenance dashboard, right? So this allows you to know how much vessels within a fleet are ongoing repairs, what are due, what is the average spend that folks are spending maintaining the fleet, cost per down, different vendors are going to use, different types of repairs going on, as well as having a maintenance schedule. Here you can see all the different dashboards that we offer. So these are just a few that we've sampled to show you exactly what's offered here. But we're going to go over to the incidents and SAR operations. So you can see active incidents, what's the average response time, how many results you did in different types of incidents, see the trend rating, the response time analysis, severity distribution, different high risk areas and what's not. And then obviously as we connect this to our VMS, we'll be able to bring a live map there. And then we also have a dashboard tracking the different vessels are under construction projects, how many we delivered, what's the actual investment, the average completion, what are the actual projects that are going on, what's not. And this is also data data, obviously, so none of this is like actual data, but this is to treat, this is so you can understand how the system's capable. And then we're also providing portals for people to be able to provide inspections data to the inspectorate, as well as being able to provide portals that would go out to the Coast Guard. So if the Coast Guard needed to check in with the Barbados Fisheries Division related to where a particular vessel is located, no more will they have to be calling Hadrien a million times. They'll be able to have their own centralized portal, put in some information, and as you see over there, they'll be able to download their own Excel sheet. Of course, Barbados Fisheries Division will have total and complete control over what the Barbados Coast Guard or any other data partner will be able to see. Right. So how does this help anybody, right? All this technology that we've built, the way that we see it is that firstly, for researchers, it will allow researchers to be able to take their research and just, always having to answer, hey, do you have this data set? To being able to go and see what is available and be able to start your research from day one. The second thing is that we're thinking about how this helps the fisheries management officials, even outside of FAS. You will have a platform which shows you exactly how you can store this type of data going forward, how you can build very interesting processes and very scalable architectures. And then for government policymakers, well, it's already going to start helping the central bank to be able to do better analysis and understand how Barbados' fisheries division is actually contributing to GDP. Because for the first time ever, you'll be able to see exactly how the different inputs go directly into a landing coming out of the ocean. Right? At least that's the very, very hope. And so we're not done yet. So our roadmap for Q3, we're doing further VMS integrations with more and shall catch so that in the actual FAS, you'll be able to see a bit of a delayed real-time mapping of the actual vessel in the ocean. We're also adding a FAS AI-powered assistance, which will improve fisherfolk access to fishery services via WhatsApp. So this will be something that you'll be able to just go on WhatsApp and say, hi, I am Joe Blow, here's my fisher ID card. It will do an authentication process. And then you'll be able to do things like requesting your owner certificate or requesting other necessary details that you may otherwise have to spend an entire day coming down to the Barbados Fishery Division to actually perform. And then for 2026, we want to expand to complete and to cover the entire marina so that we can increase our data capture to provide an even wider view of the actual data. Okay, so Intelligent Technology Inc. is a deep tech research firm focused in Barbados. We have a history of working with the government for the last 5 years. We've built and researched things with them in machine learning from image understanding and image processing all the way down to this project that we're working with them here. It's been an absolute pleasure and honor to work with the team at Barbados Fisheries Division. And I hope that our collaboration continues. And if you want to connect with us and learn more about this project, you can reach out to Shelly, reach out to myself, or if you actually just want to have access to the API, you will also be able to do that. So thank you very much. This is my presentation.
All right, that was a great presentation. Do we have any questions here in the room for either Christian is online as well as Shelly-Ann? Going. All right, I just want to tell both of you, I don't think we can see you online, but great job. I'm really excited as a Bayesian to see the progress of this platform. Shelly-Ann.
Thanks, Kim. Thanks, Kim. And we are having a good time with the stakeholders, especially building in the feedback for the dashboards. So most recently, we had a maritime safety workshop where we had members from Marine Police, Coast Guard, fisher folk, and everybody finalizing what we thought of the harmonized standard for the vessel incidents and search and rescues report should be, which would feed into the dashboard that Christian would have presented. So lots of good things happening, and also great to see the developments with our other colleagues that presented earlier. Wish I was there in Colombia, but we are still working on barrel recovery.
Great. Thanks, Shelly. Well done. I'm really proud of you guys. It's great to see the progress you've made just in such a few short years.
Thank you.
All right, we're going to keep moving right along. Next, I have Axelle. Lefaucher.
Yeah.
I hope I did better on this French pronunciation. And she's going to be talking about Trace, a 360 AI video tool to assess the reserve effect of polluted no fishing zones. All right, Axelle, thanks.
Hi everyone, and thank you for having me today. So I am Axelle Lefaucher, and I will present you today a snip of my thesis about an AI tool to assess biodiversity. So I'm from the National Marine Park of Martinique, and I'm actually the project manager of the European Project Trust. So we will go with a little context about the actual study. So TRAS is the financed project by the EU. There is like two partners into this big project. So the CEFREM, with my laboratory from University of Perpignan, and I'm working actually with the French Office of Biodiversity and the Natural Marine Park. So those two partners are working closely with the fishermen of the island to this big project. So the aim of the study is to highlight the reserve effects and support the acceptance of protected area within the territory. And today I will present you the early work we did this year with my two trainees, Kate and Matthew. So here is the Natural Marine Park of Martinique. It's representing all the EEZ of Martinique. We have several regulated areas into our parks, so reserve, fishing zones, underwater trails, and area close to fishing due to chlordecone contamination. So chlordecone is also known as capone. It is a pesticide was been used in Martinique for several years, despite its prohibition. So nowadays, we have 43% of our coastline who is close to fishing. It represents 5 areas where fishing is prohibited. It's almost 10,000 hectares and of closing fishing area for health reasons since 2010. So this background, since the closure of the area, nobody have looked what actually happened to the biodiversity into the zone. So this leads to our main question. So is ending extractive activity enough to create a refuge for biodiversity? And by extractive activity, we are talking about fishing. So for that, we decide to create and develop a monitoring method that minimize bias and maximize data collection. So our first question is, does our protocol allow us to capture the diversity of the monitoring area? So, and with that, we take the opportunity to optimize our data analysis by creating an AI tool. And for that, we need to know several things for our AI. So is AI capable of detecting all the fish in the dataset? And does AI need the same amount of input data that the reference observer to assess biodiversity? So a little bit of context about the project. So everything starts in the Bahamas at the GCFI 2 years ago, where I met a young student about Dauphin Island Sea Lab, and he was presenting several techniques, several gears to assess biodiversity. And I say, okay, I have to work with this guy because I was actually looking for a protocol, a methodology to my thesis. So then we do work together. So in March the next year, I did go to Alabama and learn technique about the 360 videos. And then he came to Martinique in August 2024 to test our new gear. So is the little beast. And this is an example of what 360 video look like. And I just want to highlight why we choose to work with 360 video, because it allows us to be really precise with the fish habitat relationship and also to maximize the data collection. So then we had the opportunity to develop an AI tool with the Natural History Museum of Paris. And now I present you the DOP, so a little name to DropCam, Omnidirectional Panoramic. It's deployable from the surface. It's composed with a rosette of 5 GoPros. It's a lightweight system under 10 kilograms, and it's a full stainless steel design with 4 pods. So once deployed, it allows us to sample 75 meters square around the gear, depending to the visibility and the obstacles in front of the camera. So for the protocols, so we deployed them on 2 study sites for 2 seasons. It's representing actually 138 unique drops of 15 minutes. It's almost more than 10 extra sampled, and we need 5 days to do all the field. So at the end, we add 690 single view videos to analyze. It's a lot of hours of analyze. So it takes us 3 weeks for 2 persons to the data analysis. So let's dive a little bit into the data analysis. So 1 drops is equal to 5 single view cameras. And to do the analysis, we just look the entire first video and we note the time of first sighting of each species into the video. And we do so for all the 5 videos for each drops. And anytime we have to update the TOFS, we actually, if we see a fish on the video 1 at 1 minute and in the video 2 the same species appear before 1 minute, we just keep the second time. So we use, whoops, my notes. So to answer those 2 questions, so does our protocol allow us to capture the diversity of the monitored area, and can 15 minutes of sampling adequately capture species richness? So we model species accumulation over time using a microlist mental model to and incorporating random variable like drops and campaign, and testing different combination of fixed effects on Vmax and Km. So the model comparison via AEC revealed that the best fitting model include location and habitat, indicating these factors are key drivers of species richness pattern. So for the reserve effect part, there is no significant difference between Fort-de-France and Le Marin, our 2 studies shown about the species richness. So the species richness accumulates quite similar. So we choose to focus on the habitat. So to determine how long it takes to reach the 95% maximum richness and whether 15 minutes of sampling is sufficient, we use habitat model only. Here, the results show that our 3 main habitats, so coral, mix, and sponge, reach maximum richness within our sample time. So just to highlight, mixed habitat is composed with seagrass and coral, or seagrass and sponge. And to go a little further in our analysis, we summarize the most frequently observed species in the dataset. So we identified the top 10 species of the 102 record based on their number of observation. So here the frequency represents the number of reflects how many of the drops we actually see each species in the dataset. And so here, our 10 species represent 41% of all observation. And those 10 species are really different in terms of size, of shapes, and color. So here is the moment that we catch up with the AR part with this question: is AI capable of detecting all the fish in the dataset? So for that, we developed a detector, actually not really me, but my trainee, Matthew was a really intelligent guy. So we create a detector thanks to a visual transformer model. So I have a quick question. Is anybody familiar with AI in the room? Raise the hand. 1, 2. Okay, cool. So I will try for the other to be as clear as possible with my level on English. So for that, okay, for AI, it's quite simple. We need data to train AI. So for that, we start with collecting a lot of data on the internet. Basically, we need data to train the model. So here we use online data and already annotate images. So here you can see the source data are the images with the blue square. So with that, we run the AI and we use a lot of images. And then with the first try, with the raw data, we actually couch 2 fish on the images, but it's because on the annotated data we use in the beginning, there were annotation error on the data. So we try to ameliorate that further and further. And then we also try with data from our own videos, and this worked really, really well. So as you can see now, we are able to almost catch all the fish on the images. So we went to 2 to almost 3,000 fish catch with the AI with the best parameter of the model. So next, we try to test the detector ability to capture the species richness. So here I was the one observer and I looked through 3 drops with 1 look at real time of the video and then 4 looks with different intervals. So basically, we took 1 image each 1 second, one image is 5, 10 or 15 seconds, and to look if the AI, how many frames the AI need to assess the same level of diversity as the observer. So there is a significant difference between the different interval experienced, and we find out the 1 second interval don't have significant difference with the real observer. So for the next work, we will probably go with 3 seconds that reduce a lot of the work to do, like 19 times more efficient by analyzing only 1.1% of the data set. So it's a good thing. Here, just to highlight that we go a little further in our analyze and modeling the time required to capture 95% of the species richness within a green interval. And this accumulation rate, this shows that the accumulation rate between intervals due to differences, and it's because there is differences between the AI eye and the human eye. So that explains the difference. So for little conclusions, we have an effective gear and methodology can detect our diversity within the 3 main habitats and have a possibility to optimize the data analysis. And we are on the way to classify them. Thank you so much for your time.
Thanks. Great presentation. Are there any questions for Axelle?
No.
Well, I, of course, have some. I just had a question. What is so I saw that it was what I appreciated about this is that it looked like it was a very simple setup. You said it was just 5 GoPros tethered down, because I've looked into ROVs, as you know. And what was the cost of that equipment? And it was great to see the time and savings and effort. So are you quantifying that for your PhD research and also going to be doing like an economic evaluation of the cost effectiveness of this approach? Because it looks pretty promising based on what you're showing here.
Yeah, thank you so much. Yeah, it was one of the objectives because I really want to create a methodology that can spread around the Caribbean with something like cheap but efficient. And actually today, I think everybody can buy a GoPro of 5 of them. And so for the gear, it was less than 400 euros to build deep. And for the GoPros, it's like nowadays with the newest, you have like maybe 100 to each. So it's really cheap, really cheap. And the second question was?
Well, the software.
Oh,
Is that expensive?
for the moment, it's free. It's only a free model on the internet. So, I forgot to say that we actually are writing a methodology paper about it. So we actually succeed to classify them. And so I hope by 2026, we will be able to have like a real tool, like a click and click thing that you can just put your videos in and get the data out.
That's great. Yeah, my other question was just, are you quantifying the cost savings as part of your PhD research?
I think because of your question, I will.
I think you really should. I think that's what people need to see is the cost effectiveness of these tools. They're generally getting easier and easier to use, and they're saving us extreme amounts of time and money.
Yeah.
And I find by getting sustainable financing mechanisms, you always have to have the economic ROI
Right.
of these innovations. So well done.
Thank you.
All right, and our last presentation is from Hanna-May Malahel. Hopefully I didn't bash that one up too much. Mapping nitrogen inputs into the Grand Cul-de-Sac Marlin Bay of Guadeloupe using an isotopic approach. Thanks, Hannah.
Hello everyone, buenas tardes a todos. My name is Hanna-May Malahel. I'm a PhD student at Université des Antilles, and I am very pleased to present my research here at the GCFI. Today I will show you how we map nitrogen inputs in the Grand Cul-de-Sac Marin Bay using an isotopic approach. So this project is part of a study covering the entirety of Guadeloupe, but today I will focus only on the Grand Cul-de-Sac Marin Bay. So if you were here at the last GCFI, you already know about Guadeloupe Island, located in the Les Antilles, where the last meeting happened. But do you know about the greatest bay of Guadeloupe, the Grand Cul-de-Sac Marin Bay, located to the north side of Guadeloupe? The Grand Cul-de-Sac Marin Bay is home to the largest reef barrier of the Les Antilles, home to many reef communities, seagrass beds, and mangroves. This mosaic of ecosystems offer great opportunities for economic matters such as fisheries and blue tourism. Unfortunately, this bay is also the receptacle of many land-based pollution that threaten the resilience of these ecosystems of high value and also threaten the sustainability of this economic activities. Among them, nutrient-rich inputs from wastewater, urban, agricultural runoff, harbors activities are known to favor algal overgrowth that bring the process of algal coral shift. The scarp sources influences of mineral pollution in the Grand Cul-de-Sac Marin Bay is still unknown. So for this study, we try to answer this question. What are the sources and spatial distribution of nitrogen inputs in the Grand Cul-de-Sac Marin Bay, and how do they relate to reef health? So to investigate the mineral pollution in the bay, we collected macroalgae every 2 kilometers between 3 and 5 meters depth. The macroalgae was Dictyota. This is a brown algae distributed across the entirety with a great distribution on Guadeloupe Islands and are known to absorb nutrient pollutions, especially nitrogen. So we analyze nitrogen isotopic composition expressed with delta 50N. Delta F50N is a ratio between the heavier and lighter nitrogen from my samples compared to the same ratio from the standard. And the standard is atmospheric air. So basically, delta N15 value less than 3, which means a natural or fertilizer sources. Meanwhile, a higher value, so more than 3, will means anthropogenic sources. We also map land-sea pollution factors using smart map on QGIS and the distribution of delta 50N using interpolation EDW on QGIS. So here we have the first map of the distribution of delta N15 values in the Grand Cul-de-Sac Marin Bay. So what we have here is a gradient from blue to red, so 0 per mil to 4 per mil. The sites are represented with the black dot, watercourses with blue lines, coral reef, dash pattern, seagrass beds, dot pattern, and finally, mangrove in pink. So the maximum value of delta 50N has been reached near the channel with 4.28 per mil, and the minimum on the coral reef barrier with 1 per mil. So in the next few slides, we're going to see what are the factors of this enrichment. But right now, we can already say that the minimum 1 per mil on the reef barrier is natural influences. Here we have represented the wastewater treatment plants in triangles. So red triangles for non-compliant and blue triangles with compliant. Delta N15 values are high near wastewater treatment plants, and it's about 57% of wastewater treatment plants which are not compliant around the bay, according to the latest report from 2022. And to have an overview on Guadeloupe, it's about 56% of wastewater treatment plants which are not compliant. Here we have represented industrial area and the harbors. So as you can see, our industrial areas are very minimum on the map. It's represented with the in orange. So we can say that there's no influence on nitrogen enrichment. It's not significant from industrial areas. But if we looked at residential area represented in gray, we can see that they are concentrated on the coast. And one thing important to note here is that the rainwater collection network in Guadeloupe is non-existent, which means everything flows into the water without any treatment. Here we have the agricultural plots. They are also concentrated across along the bay and near water courses. So as I said before, there's no rainwater collection network. So the water courses have the receptacle of the runoff that goes directly into the sea. And one thing, one particularity about agricultural plots using fertilizer value, delta N15 values are very low. By low, I mean between -3 to +3. And that's why on the coastline, we can see there are sometimes low values. So to conclude, the nitrogen N15 enrichment areas was identified near wastewater treatment plants, urban areas, river mouths, and channel. The highest values was near the channel, and the channel is going through the biggest city of Guadeloupe, which is Pointe-à-Pitre, with a high density of population, urbanization, and also connected to the other bay of Guadeloupe on the other side, and currents flows between the channel. These results are consistent in the tropical context, which means oligotrophic waters and efficient ecosystems. When dissolved nutrients are available in the water, there is an efficient uptake that happens. For the perspective of the study, processing of the data from the dry and wet season is a cooperation between them. And if you'd like to learn more about this project, please feel free to scan the QR code on my poster to see a short documentaries on YouTube about this project. And you can see how we collected data on the field, some scenery of Guadeloupe, and some more information about the analysis. Reaching the end of this presentation, thank you very much for your attention. Gracias a todos y puede hacer sus preguntas.
Great. We have some questions. Henri.
Thank you for the presentation. So I have a question. Obviously, you can tell the origin of the nitrogen based on the isotopic signature, but can you use your data also to tell how much nitrogen is getting into the system? Have you thought about looking at NC ratios, for instance, in the tissues of the algae or something along those lines to figure out whether there's too much nitrogen getting into the water? Is that something that you can look into, or I don't know, something? Have you given it any thought?
No, I'm sorry, but I didn't hear the first part of the question.
The isotopic signature tells you where the nitrogen comes from in terms of whether it's wastewater from human origin, but it doesn't tell you how much of it is getting into the system in terms of runoff, for instance. So you may get nitrogen that comes from agricultural practices, which has a different signature. and nitrogen that comes from wastewater, from human activities, which has a higher, a higher signature. But can you tell from your data how much of it is getting into the system? That's what I'm trying to ask.
So if I understand well, the question is you want to know how much the influence of wastewater compared to another sources or
Yeah.
projected sources.
Yeah.
So the threshold of delta N15 values are, according to the bibliography, are sometimes the same. For example, wastewater treatment will vary in coastal environments between 4 and 38, depending on the duration. And so this is wastewater. This is the highest you can get. But for example, another example with wastewater, urbanization runoff, we have an also high value, and also starting from 3, but according to the literature, it stops at 7, so 3 to 7 for urbanization. So to discriminate the factors, I think it's very important to consider what are the sources near the point of collections. I don't think you can just look at this value and say, this is it. They need to have a better investigation on what are the costs, where are the outfalls of wastewater treatment plants, the currents too, and so abiotic factors to play to the value.
Okay, thank you.
Great. Okay, we got one more here.
So you have a lot of seagrass there. How do you think that's taking up some of the excess nutrients compared to the macroalgae?
I don't hear the question because the volume is low from when I'm here. Can you repeat, please, with closer?
Oh, sorry. I can't hear. I'm old. You've got seagrass and you've got macroalgae. I'm assuming seagrass is taking up nutrients as well. Do you think the values in the macroalgae are lower maybe because there's so much seagrass there? I mean, are they sort of competing for what's available?
We collected macroalgae. It's only on the reef. So yes, there's a lot of seagrass bed patch, but we paid attention to only collect on the reef, coral reef, not where they were fighting with seagrass.
Okay, great. That is the end of our session then. Thank you very much for your talk. And
Thank
I
you.
would ask that all the other presenters please come up here. And thanks everybody for a great innovation section. So we're going to take a break right now. I think we caught up on time. We're 3:58. So please make sure that you come back promptly at 4:30. So all the presenters are going to come take a photo if the presenters are still here. I couldn't hear that myself. Oh, the auction is closing soon. So go in right now, make the final bids. This is your last chance. We're coming to a close. Thanks, everyone.
That's my dream. That we're never gonna survive unless